Welcome back, everyone, to another session of this webinar series on scientific machine learning. I'm excited for this last talk of the series on molecular machine learning for drug discovery. And today we have Mario Krenn from the University of Toronto presenting. Our session chair will formally introduce Mario in just a bit. And in the meanwhile, let me introduce our session chair for the day, Professor Rafael Gómez-Bombarelli, who's an assistant professor of material science and engineering at MIT. Rafael joined the MIT faculty in January 2018. He received a BS, MS, and PhD in chemistry from the University of Salamanca in Spain, followed by postdoctoral work at Heriot-Watt University and Harvard University. His research combines first principles simulation with machine learning on theoretical and experimental datasets to accelerate the discovery cycle of novel practical materials. His group has been focused on addressing the role of molecular transformation in materials discovery in several areas, including designing the stability in advanced materials, also catalyst design, and novel and replacement chemicals. So, Rafa, very pleased to have you chair the session. Thanks a lot and welcome to the session. Over to you to introduce Mario.
Thanks very much, Dilip and Venkat, for having me. I'm almost ashamed to say, we'll catalyst design in front of Venkat, but we're trying our best. It is truly a pleasure to introduce Mario Krenn, who got his PhD in Austria at the Zeilinger group, doing old school hardcore quantum simulations, like the rigorous stuff. He did a little bit of a postdoc there as well. Then he moved to Toronto with the Erwin Schrödinger Fellowship to work under Alana Spuru-Guzik, who I'm hoping lots of you know, and I know also personally. And from that collaboration between Mario and Alan, has come all this exciting work around selfies and sequence representations for molecules that I find extremely, extremely appealing and sort of really a watershed moment in molecular representation and generation. So I'm excited to hear this talk, and with this, I'll hand it off to Mario to tell us about it.
Cool. Thank you very much. Let me share my screen. Let's see. Okay, can you see the screen?
Yes. Yep.
Okay, cool. Okay, thank you very much for inviting me. So I will talk today about selfies. That's our new representation for molecular strings that we use and used recently extensively in machine learning in chemistry. So I work in the group of Alana Spuru-Guzik, and that work has been done together with a number of my colleagues, in particular with Florian Hese, Akshat Nigam, Pascal Friedrich, and of course Alan, who was the team with whom I kind of invented selfies, or I had the first ideas. And then since that time, a lot of development has been done in the last year. Specifically, I want to mention Alston Lowe, who has basically completely rewritten the selfie GitHub repository, made it much nicer, with the help of Seyon Chitranandra. And Cynthia Shen and Sagi Apple have worked with me on the deep dreaming part that I will mention later. And Luca Dieter has worked on curiosity-based reinforcement learning using selfies, which I will, if there's still some time, also mention briefly. And Robert Police, who is basically a chemistry advisor for selfies. Okay, my background is not chemistry. I'm a quantum physicist, and actually I started as an experimental quantum physicist. And during during my PhD, we were trying to build experiments that can investigate some of the fundamental property of quantum systems, for instance, quantum entanglement. And at some point, we wanted to create some very specific, very complex entangled system in the lab to observe its properties. And at that time, usually when we want to do the task, we sit down and we write down with our intuition all of the experimental setup and think about how it could work. And usually we find solutions rather quickly. But in that case, several years back, it was different. For several weeks, my colleagues and I, we were trying to find actually an experimental feasible way to produce that quantum system. But we were not able to do that. And then after some time, I thought, okay, that's very curious. And I tried to come up with a different method to design quantum experiments, not with human intuition, but I thought about how computers can help to design experiments. And that was actually a very fruitful approach. It was not done in that field that I was working on before. And then after that, it became quite a fruitful approach. And several of those experiments that have been designed with my algorithms have then actually been implemented in laboratories and have led to new observations of new quantum effects, quantum interference, observation of new entanglement structures, and so on. So this was I found this very exciting, and I wanted to learn more about how could we use AI in the field of designing quantum experiments and advancing physics. And I've looked into the literature, but it seemed that this question was basically not addressed in physics. So there was no way how I can use some knowledge from others and apply it in quantum physics. But then I found that actually in the field of chemistry, the question of designing new structures emerged, and a lot of work has been done in that field. So in particular, at the time, I found this very beautiful paper where the authors have shown how to design new molecules using deep neural networks. That was a very appealing article for me. I found this absolutely fascinating. And of course, it's the first author is our session chair, and I'm very happy that he is now here in the audience. So that paper influenced me a lot, and also then this other review that shows, okay, there are several different methods how you could actually use deep learning to design new molecules. So at that time, I thought, okay, I definitely want to learn all of the things that the chemists know about designing new things using deep learning. And that was the reason why I decided to go away from a field of expertise, physics, join a computational chemistry group of Alana Spuru-Guzik, try to learn everything that chemists have developed and bring it back to physics. Okay, so when we think about the designing new molecules, this is often called inverse design. So inverse design comes in the context of structure-property relation. Here, structure could be a chemical molecule, or it could also be a quantum experiment or any other physical structure. And the property can then be calculated using, for instance, Schrödinger equation, or can be measured with some experiments or with some simulation. So this is the direct path. But inverse design is kind of the opposite way. You're given a certain property, and you want to find the structure that corresponds to this property. And this question is tackled with a lot of different methods, for instance, evolutionary strategies, but also deep learning methods like the variational autoencoder that was pioneered by our session chair. So if you want to use a computer to design new molecules, a very first question that you want to ask yourself is, how can you represent the information of that molecule? And there are basically two different ways how to do this. The first one is the so-called adjacency matrix-based representation. And the second one is the string-based representation. So in the adjacency matrix-based representation, this is also sometimes loosely called graph-based method, but both representation can represent graphs. So in this adjacency matrix representation, you take a vector that stands for the nodes in your graph. And then you have a matrix that stands for the edges that are drawn between your nodes. So with these two objects, you can represent a graph, and in this case, a molecular graph. The opposite or different method are string-based representation. And here, basically, the most famous string-based representation is called SMILES. This was invented in the late 80s and has since been basically the most important representation in computational chemistry. The idea of representing a graph or a molecule with strings is very clever. So what you do here, you take your graph and you draw kind of one main string through the graph. So here you see this green string. You can think of the main string, and you just take your atoms that follow each other and write them in a list. For instance, here you have a carbon, nitrogen, carbon, carbon. This is here carbon, nitrogen, carbon, carbon, and so on. So with this linear chain, you already have much of the molecule represented. So then there are two more things that you need to take care of. There can be branches, and in SMILES, you represent branches as brackets. So at the third atom here, you make a branch. It means you write the bracket, and everything that is in the branch, you write into the bracket. Here you have one carbon atom, so you only write one carbon. The second thing you need to take care of are, you can have connections from a later atom in your chain to a previous atom. So basically you can form rings. Let's look at this ring here. You identify this additional edge by putting an integer at the location of the first and the initial and the end point of this additional edge. So in this case, we have a symbol 2, and you know you interpret this as one additional edge in the graph. And with these two additions, you can basically represent all chemical or molecular graphs. So in some way, I think that string-based representations are actually theoretically extremely powerful, because you can think about strings. Strings can encode arbitrary computer programs. So a computer program is basically just a string that is interpreted in some way. So string-based representations are basically arbitrary, computationally powerful, or as powerful as the best classical computation algorithm. And in many cases, your molecular graph, or in general graphs that come from the natural sciences, are not just blind 2D, but have a lot of additional structure. For instance, in chemistry, you can have additional 3D structure or different type of bonds or physical constraints and so on. And those you can encode quite simple in string-based representation, which becomes kind of challenging in this graph, in this adjacency-based matrix representation. But potentially some people in the audience might have different view on this. But because of this, I will focus now on string-based representations. So let's go back to this really cool invention by our session chair. So they came up with a way to use variational autoencoders to design molecules. And that works in the following way. You take a molecular graph and you write the molecular graph in a string-based way as smiles. And then you take the smile strings and you encode it into a continuous representation called latent space. And afterwards, you take a decoder, other neural network that can take a point in the latent space and decodes it into a molecular graph. And in the training, you basically in some way learn an identity transformation. But the cool thing is, here in the center, you have a continuous and therefore differentiable representation of your discrete structure. So you kind of go from the discrete to a continuous representation of a molecule. And this is what you can exploit for design. So here the idea would be that you take a third neural network that can take a point in the latent space and predict the property of the graph that corresponds to this point in the latent space. So this is a property prediction neural network. And with this property prediction, you can then continuously move through the latent space. That's the idea. You can continuously move through the latent space, and in order to maximize a certain property, all in differentiable way. This is the main idea of this very beautiful paper. Now there are a number of problems, and one of the problems is that smiles, the output, is in many cases, or can be in many cases, invalid. So that means it does not correspond to some meaningful molecule. And that can be because of two reasons. Either, one reason could be that there are syntactic mistakes. For instance, imagine you have a bracket and that bracket is not closed. So that doesn't correspond to any meaningful molecule, doesn't describe a meaningful molecule. Second, there are there can be semantic mistakes. And those are mistakes that come from constraints from the domain. For instance, in chemistry, we know that the oxygen can only bond to two other atoms. That means if the string would say, okay, hey, bond the atom one time with this carbon and two times with this carbon, you would have 3 bonds. That doesn't make sense physically. That doesn't describe any physical realizable molecule. So here you have semantic mistakes. And those mistakes happen actually quite frequently. If you look at the latent space of a variational autoencoder that is trained on smiles, and then you scan through some of the dimensions in this latent space, you see that many of the molecules that you would decode, or many of the strings that you would decode, correspond to invalid strings. So here red is invalid, green is valid. So you see huge regions are basically invalid. So the whole idea of this differentiable optimization kind of gets quite difficult. So people have come up with a number of different potential solutions, and there are basically two different ways. One of them is that you try to modify your model such that it fulfills those constraints, such that those systems cannot be created. Those invalid systems cannot be created. Also using filter technique and so on. So the problem is that this is kind of model dependent, and if you want to apply some optimization using another model, you have to do the work again. A different, kind of more fundamental solution is changing the representation itself. So this has been done the first time roughly 2 or 3 years ago, using where the authors defined a new way to represent molecules. They call it deep smiles, and they identified a number of problems of smiles and fixed them in their representation. The problem is that, okay, the good thing is this actually fixes a number of syntactic mistakes. Unfortunately, still other syntactic mistakes still exist, and especially it doesn't take care at all about the semantic mistakes, so those domain-dependent mistakes. And this is where our representation comes in. We define a new way of writing down molecules as a string, but in a way that it is 100% robust, so that it's always produces valid outputs. And the main idea behind SELFIES is that you use a derivation system that is inspired by theoretical computer science, by ideas of formal grammar or formal automatons, in such a way that it satisfies both the syntactic and semantic constraints all the time. And you can think about this representation in such a way that every combination of the letters of the alphabet gives a meaningful molecule. If you think about natural languages, this would be a language where every combination of letters in the alphabet gives a meaningful word. Let's think about the English language, alphabet from A to Z. Every combination of letters should give a meaningful word. That is definitely not the case in English, but this is what SELFIES is in the chemical domain. Okay, so now let me briefly explain how we fix those mistakes. I mentioned there are two problems, syntactic mistakes and semantic mistakes. So first, the syntactic mistake. The problem of the syntactic mistakes is that in smiles, you represent the start and end point of rings or branches at two different locations. The problem is now that the model could maybe start a branch, but then forget to close this branch later on, which leads to a mistake as I've shown here. Now what we do is we define the ring and the branch at one location. We do this by saying, okay, at this location, I want to make a branch, and the branch has this size. So that means the branch is well defined, and it is defined at exactly one location. Let's take this molecule as an example. We have 4 carbon atoms. Then we say, okay, 1, 2, 3, 4. We want to create a branch here, and the branch has size 1. And the size 1, that is the 1 oxygen, and we continue with some carbons. Now, we cannot write the size as a number, because otherwise, if we would use that number symbol without the branch symbol, this would again lead to invalid structures. So we use a trick. We actually define the next symbol after the branch symbol. We don't interpret it as an atom, but we interpret it as a number. Whatever it is, we interpret it as a number. And we use this base 16 system, where here carbon stands for 0 and so on, and up to P, which stands for 15. And we say, okay, the next symbol gives the size of this index plus 1. So in this case, the size is 1. Index of C stands for 0. 0 plus 1 is size 1. So this carbon says there's a branch with size exactly 1. This 1 is this oxygen that comes after the number. If we take now a system that has a branch of size 3, we say, okay, size 3, look, 2 plus 1, this is the ring 2 symbol. This is now interpreted as a number, stands for the size 3, those three oxygens. And very similarly, we can do exactly the same in rings. We define a ring and say, okay, we connect to the 7th atom before. And here 7 is 6 plus 1. This is this branch 2, 1 symbol. And that defines the ring at this location. So we fixed the syntactic mistakes. Second, we have semantic mistakes, those domain-dependent mistakes. And here we exploit not as formal automatons, these derivations, where the state of the derivation kind of carries a tiny amount of memory. That memory stands for the number of bonds that are still allowed to be created. For instance, this X1 says, okay, I can still create one bond. So the derivation of that symbol is state dependent, as it is always for formal automatons. And here, a very simple example, we always start in the X0 state. And here I want to translate this selfies. So F in X0 stands for X, and we go to the state X1, F, and we go to state X1. Now we are state X1. The next symbol is double bond C. Double bond C, X1 produces a C, and we go to the state X3. Again, we go to the state X3. Double bond C produces this symbol. We go to state X2. X2, the next symbol is the sharp N, produces a double bond N, and we finish the derivation and we get a valid molecule. So those are now the two solutions for semantic and syntactic issues. If we now look at what happens if we take a string and change a number of letters in the string. If you do that in smiles, as mentioned before, many of those do not correspond to valid molecules. They are either syntactically or semantically wrong, or in the worst case, they are wrong in both with both respects. But if you do this with selfies, the graph changes, but it still always stays a valid molecule. We can do this more systematically and analyze what happens if we change a lot of bits in a lot of symbols in the string. If we start with some valid molecule and we change a lot of symbols, for instance, if we change 10 symbols, in smiles we get less than 1% validity. Deep smiles less than 5%. In selfies, 100% valid. Now we can apply this to our variational autoencoder. And we see if we train variational autoencoder with selfies, it's 100% valid. But not only that, also it has a higher reconstruction accuracy than if we train the same models with smiles or deep smiles. And importantly, it has a much higher diversity than if we train it with smiles. And we can not only use this representation for a variational autoencoder, but it's totally independent of the model. So we can also apply it to generative adversarial networks. And here, again, the diversity of the GAN is much higher if we train it with selfies. So this was the picture that I've shown to you before, the variational autoencoder and kind of sampling through the latent space. This is how it looks with smiles. And this is how it looks with selfies. Every point in the latent space is valid, and that should simplify the continuous optimization of molecular properties. Now, the VAE is not using selfies not only more valid, but it also has a much higher diversity. And this diversity is in some way the resource that we have for our optimization. So the diversity of the molecules is kind of what we could potentially reach. And the more we could reach, the more potential we have to reach some good molecules. And in a test, we have seen that a VAE with selfies encodes 2 orders of magnitude. So here there's a logarithmic scale, 2 orders of magnitude more molecules than if we would train it with smiles or with deep smiles. We have used now selfies in a number of applications, and the first and most direct application is in a genetic algorithm that has been spearheaded by my super talented colleague Akshat Nigam and was published in ICLR last year. And here, just by changing from the smiles to the selfies representation, we have a genetic algorithm that outperforms most of the other models. But we have also applied this technique now to deep learning, based design methods. And specifically, I want to talk about this one work that has been mainly done by my colleague Cynthia Shen. Here, we were inspired by the idea of deep trimming in the field of vision. And the idea of deep trimming is that you take a neural network that can classify your images, and then you give it a new image. But instead of letting the neural network classify the image, you take one neuron, for instance, one in the second to last layer, and you try to maximize the output of that neuron by changing the picture. And in that way, you kind of can get a rough idea of what this neuron is looking for. So here are two examples. In the middle example, it kind of looks for straight lines and some geometric shapes. In the second example here, this was done, optimized a different neuron. It kind of looks for vortices and so on. So something real. So you can kind of learn what the neural network is looking at. And with the steep trimming technique, you can not only look at those output, but also create absolutely fascinating pictures. So you input some picture that you like, and then you optimize one of the neurons, and it changes the picture. And it could, for instance, look like this very dreamlike image. Or here, Mona Lisa with a bunch of dogs inside of her. Very disturbing picture of the royal family. Another Mona Lisa with dogs, then dogs with dogs, which is also pretty cool. Or Van Gogh with some chicken and snakes coming out of his face. So you can create very fancy pictures with this. And we wanted to use this technique in chemistry. So what we have done is it consists of two steps. In the first step, we take a neural network and we give it a molecule, and we train it to predict a certain property. Now, if it trained the property well, we stop and go to the next step. And in the next step, we freeze the neural network parameters, and we start with a given value that we want, a property of the molecule, and then we ask, how would you change the initial molecule such that you go into this direction? For instance, if this value is far outside of your data set, how would you optimize in the upper or lower direction the property that we are interested in? So in this way, we kind of in a very direct way, in a differentiable and very direct way, can optimize molecules. And this is made possible, or at least was much simpler, by using surface representation, because here we don't need to be scared that the model gets stuck in some invalid valley of smiles. So here, every output that we got was valid, and also very interesting, we can then follow the intermediate steps, and every intermediate step is also valid. So we can see how the model changes, which I come to in a second. So the input of our model was, so we took a molecular graph and we transformed this molecular graph into a selfie representation. And this we transform into one-hot encoding. And then, very important, we needed to add noise. And this can be understood quite simply. In this backward training, what we do is we start with a molecule, and if the molecule is represented with one-hot encoding, it continuously changes the one-hot encoding into some other type of matrices. But if the neural network has never seen continuous values, if it has only seen zeros and ones, it has basically no idea what this means and gets stuck very easily in some minima and doesn't make sense. So it's very important to train the neural network for this deep dreaming with noisy one-hot encodings, such that in the backward training, it kind of understands what these intermediate states mean and can pass through them. So here's one example or two examples of such a process. We take on the y-axis, the brown one is the physical property, which is kind of the hydrophility of the molecule. It's not so important here. We take a molecule and we say, okay, how about you try to minimize this property? By minimize, we mean we set the goal value to a very low property. And what happens is we train over a number of epochs and the loss decreases. And while the loss decreases, the one-hot encoding changes. And it then it changes continuously, and then it stepwise changes the molecule from this initial structure to a different structure. And this is what happens. There are 5 or 6 intermediate steps until it reaches this final good molecule, which actually has a much lower log p value than the initial value. And this is not cherry-picked. This is basically what happens most of the time. Here, another example is when we train into the positive direction. We start with a molecule and we see it continuously decreases the loss until it's saturated, and while doing so, it increases in a discrete way the property of the molecule. And this is really made possible to see all of these intermediate valid structures by the use of selfies. And here we can take a huge set of molecules and one time take the data set, train it into the negative, and one time into the positive direction. We see that actually the distribution split and go outside of the data set. So not only it produces these edge molecules, but it produces new ones that haven't been in the data set. A cool thing is now, exactly with the same idea what the machine learning people in vision have done, you can try to understand what the neural network is learning, and that is by giving some certain initial states and look at how the neural network tries to modify the structure. And in this case, we try it into a positive and a negative direction. In the positive direction, it makes the molecule larger, which my chemistry friends can explain, so it seems to make sense. And in the negative way, it makes the molecule smaller and tries to go to water, and that also makes sense because the property is related with the hydrophility of the molecule. And so we can look at how the network is actually performing its transformation, and not only the last step, but also all of these intermediate steps. So we can kind of get design principle, computer-inspired design principle from here. Okay. Probably I will skip this application to curiosity-driven reinforcement learning. If somebody's interested in, I can talk about it briefly later. But I want to mention this specific research, which has been done by, not by our group, but by a different group, which just absolutely blew my mind when I saw this. Here the authors have solved the image to string translation task. So the question is, given the image of a molecular graph, how does the string-based representation look like? So you have a molecular image. How do you go to the string? And they used CNNs for the encoder and RNN for decoder, and they use actually a lot of techniques from image augmentation and natural language processing. So, but the very cool thing is that they have seen that this model works better if they use deep smiles instead of smiles. So deep smiles is the slightly more robust representation. And then in the conclusion of the paper, they write that actually selfies has outperformed deep smiles. So in some way, this model works better if it uses a more robust representation to write the molecule. So here in no way there's a connection to this robustness, at least not in a direct way. So it seems like that this model finds it easier to understand and to write selfies than smiles. And this is really, really strange because I really don't understand why that is. And okay, it could be an outlier, but the same group has then performed a different experiment where they translate between two different strings. One is the smiles or selfies, and one is the IUPAC. You can see here one example. And they translate between those two strings in both ways. And in both ways, they have seen that selfies outperforms the other representations. So those models, again, find it easier to use selfies for reading and for writing these new strings. And I really don't see why that is. And that must have something to do with machine understandability, which I don't even know what that would mean. Why does this machine find it easier to use that more robust representation where robustness doesn't go into this task at all? Okay, so I'm basically out of my time, so let me go to a conclusion. I've shown to you our new representation, which is simple. It's a string-based representation. It's 100% robust, and it's basically model independent. You can use it for variational autoencoders. We've used it for GANs. We've used it for genetic algorithms, for reinforcement learning. Other groups have used it for natural language translation tasks. And you can think about the language in such a way that every combination of letters in this alphabet gives a meaningful molecule, chemical word, which doesn't exist for normal spoken languages. And here are the resources. Here's our paper that we published last year. We have a lot of resources at our GitHub web page, also a blog entry, which basically I followed during my talk. Selfies is pip installable, so it's very easy to use. And if you like selfies, please give a star please give a star at GitHub because we have a friendly competition with the other repo, and we definitely want to win that competition. So thanks for supporting Team Selfies. Okay, now the other outlook. Selfie is not only a representation for molecular strings, but you can think about it as a representation for arbitrary semantically constrained graphs, and where the semantic constraint comes from the domain that you're working at. And in chemistry, there's constraints come from, for instance, from the number of bonds that atoms can have. You can also think about it in physics, in quantum physics, quantum experiments, which was at my first slide, my motivation to start in this field. You can use selfies also for quantum experiments. And in the supplementary of our paper, we actually wrote down the grammar of quantum optical selfies. So that would allow for a lot of cross-domain learning, where actually you have one representation. And the grammar rules you can basically could get out from a data self automatically. You don't have to write down the grammar. So this could lead to a lot of really interesting cross-domain learning mechanisms. And my other question that I would find very interesting to understand more about, and also very interested in what you're thinking about, is what does it mean that a machine understands some representation better? How can you test this really, and how can you design new representation? Because in string-based representations, we have so much degree of freedom. I mentioned that theoretically, this is you can see it as a computer program, and computer programs, you can write very complex codes. So you have so much degree of freedom. If we have a way to think about what machine understandability means, we could potentially find much better representations. And then there's also the question, if we think about machine understandability, what is human understandability of a representation? How would you test it? Would you just take 100 people and give them different representation, or can you do something cheaper? And can we learn something from the human to build up some good machine understandable representation for semantic graphs and molecules? And with this, thanks a lot. And I'm finished with my talk part. If I would be very happy to take questions now or also later, whatever you prefer.
Thanks, Mario. That was really, really good. We've got an array of questions. I don't know if you see them, but they've been accumulating. They range from people that are that sort of want to pull out a little bit of controversy to a little bit more technical things, and another is a code review or sort of a code demo part of this. So I don't know if Dilip has any thoughts about how to time it.
Yeah, I think Rafa, we could take just a question or two, simple questions now, and go over to the code session.
Okay, I think there's a couple that came in that are, I think, easy to clarify. One is from Kian Po. They're asking about how do you handle stereoisomers and chirality?
So basically, we can represent everything in the same way as Smiles does it. So in Smiles you have this for stereo centers. You have these @ symbols. You can use exactly the same prefixes in selfies, and it still remains valid. Also, you have this how this symbols called. These symbols, I don't know the English word for that. You can use those in selfies as well for prefixes to add a little bit more of 3D structures.
Excellent. And then there was another one that I think asking about from Cyril, asking whether the interpretability in image to string might be coming from just the density of information in the string. So maybe debugging all of image to string is hard, but are selfies longer, shorter than smiles, sort of same length? Do you have a feeling about that distribution?
Yeah, they have very similar length. So basically the same length. The density could be in some of the directions, but for instance, if you have selfies to this IAPUC, to this other representation translation where you read selfies to this other, I don't see how the density really comes in, and it also outperforms the other representation. So that would certainly be interested to see whether the ratio of density can explain this one direction, but then you still have this other direction where you read selfies, and it seems to be easier to understand.
Thank you. There is a couple that are brewing from both Chris Rakaukas and Ole Isayev that are going to take some discussion. So I'll ask you folks to stay tuned because it will be a really exciting conversation. But in the interest of sticking to the plan of doing a little bit of code inspection now, we'll put those on the back burner for a minute.
Sure.
So we're gonna lose. Let me, if you don't mind, Dilip, for a second, we're gonna lose Chris, and he had a couple of really good questions that I would love to postulate, and maybe we can move the code to make sure that we get this one out. Chris, do you want to maybe, if you want to say it yourself, and maybe give us a merged version of the two questions you had, which I think are related to one another? If Dilip can put you on the spot.
Yes. Yeah, I just promoted him to panelist. So Chris, whenever you're ready.
Yeah. Sorry about this. I do have to take off at 12, but yeah, it's really curious, right? That you have this representation that allows you to do continuous optimization on this space, right? But when you're doing continuous optimization, you're kind of like going between things, right? So like what is that distance then between different molecules, right? Like molecules that are close in string representation, like is like is that corresponding to some real distance that you can kind of look at geometrically, and when you have a superposition between these, right, when you have like things that are like where you have the one hot vector with some randomness in there, like does that correspond to an actual molecule or something, or how would you interpret that actually physically?
Yeah, so the second question about these boundaries where the one starts and the other ends, you need to think that this, which I didn't mention, this translation is in some way probabilistic. So it's a variational autoencoder. So it's not a deterministic. It's not a deterministically that you get every time exactly the same structure back. But here, so for your first question, how do these molecules change continuously? You can look here at those structures. So let's take picture one. Here we have one molecule. How does it change? It introduces here a nitrogen instead of a carbon. But this is a very small change. Also, it looks like, okay, here it does a bit more radical change. It adds a nitrogen here, or no, it cuts out those two atoms. So they're not totally random changes, it seems, but they are also not super continuous or not one by one. So you can have larger jumps. But this is actually very interesting. I would be very interested to understand this in more detail, how the latent space is built up and how you scan through, or in this case, how is it in this case? Actually investigating how those transformations happen in all different directions. We didn't look too much into this, except of those pictures.
Yeah, I guess like another question would be, would there be another representation that, more closely puts the distances, so that way, like a small change in the string is more closely molecularly related. And, are one of the other representations more like that? Or, is there something in the middle that has like no jumps, but also only represents real molecules?
So as far as I know, SELFIES is the only one that is 100% robust. And I was mainly considering smiles and deep smiles. There's some older representation, but they are definitely not graded for robustness. It would be it could be one goal to define a different language that has not only robustness but also this closeness. As I said, there's so much degree of freedom. If this is a useful target, pretty sure that this is possible. I don't know whether this language is then easy for machines to understand, but it's definitely possible to write it down, because you can write down programs in a string. So you should definitely be able to write the target that you just mentioned. I don't know on the spot how to do it, but it should be possible.
Thanks very much, Chris and Mario. On a quick note, I would say that in my experience, these approaches learn structure similarity, but they don't learn property similarity at all. So 2 points in the latent space can have very, very, very similar structure and very different properties. So the model really struggles with doing property. It doesn't learn structure property relationships, which one would think are sort of an inherent part of chemistry. So kind of the disentangled properties in an interesting way. But lots of things to talk about. If Dilip sort of wants to MC us into the code review part, we can do that for a little bit, then jump back to questions.
So I would show first how to use selfies, and then very briefly go to the main points of the deep dreaming code, if that's fine. Should take 10 minutes maybe. So as I said, Selfies is super simple to install. You just write pip install Selfies. You don't need any other dependencies. I think we need NumPy, but that is the only thing. So when you have this, you import it, and here we have 3 molecules. And to translate the molecule to Selfies, you just write Selfies encoder of the molecule, this, and that's it. So let's run it. Our first molecule has 3 carbons, and here I want to show how the branches actually work, because I got this question recently. So here we have a branch, and it says that the next symbol is interpreted as a number. Now this ring 2, we can check ring 2 means 2. I said before the branches are size of index plus 1, so it's size 3, and that is exactly what this molecule is. So then we take the next 3 symbols, these 3 nitrogen, they are now in the branch, and then we continue with the oxygens. And here this is a different molecule where you have a double bond into the branch. So in that case, we have to use a different branch symbol, because at the branch, you kind of have to decide how many of your bonds will you use in the branch and how one will you use in the main string. So this second index of the branch symbol tells you exactly this. How many bonds will you use in the branch? In this case, this molecule has 2 bonds, and therefore we have a 2 here. Here, this molecule has 3 bonds, therefore I have a 3 here. And if you would have branches that are longer than 16 symbols, because we can only represent 16 numbers, you would write branch 2. And branch 2 says, okay, the next 2 symbols are now interpreted as a number together in base 16. And you can also do this for branch 3, so next 3 symbols. And yeah, you can, no one has complained that this is too small, but it would be very easy. Probably we have edited actually recently to do this dynamically. Okay, so this is the translation from smiles to selfies. Very simple, selfies encoder. And now the translation back is equivalently simple. We use this encoded selfie, and it's just decoder, and we write, this is the selfies, and we get back to our original molecule. Here also the length of the selfies and the split into the selfies token is done with inbuilt functions that are written in the main page of GitHub. And now here this is something quite useful if you want to use selfies for generative models. If you have a data set, our data set has 4 molecules. You can create the alphabet, the selfies alphabet from it. We do this by splitting each of the atom into its tokens and collect all of the unique ones. In the end, we add this no operation token, which is used for padding purposes. So our alphabet has this 5 symbols. Yeah, now we take one of the symbols, and we can translate these symbols into a one-hot encoding with a inbuilt function, into a one-hot encoding and into a index-based encoding. So we're looking at this COC. Okay, so this is our atom, and the alphabet. This is our alphabet. So COC stands for 131. So 131 and 44, which are the no operation padding symbols, because the largest molecule in the space has in this data set has 5 atoms. Okay, so this is basically how you would use it. Most of the functions that you need are inbuilt, and they are written at the main page of the GitHub page. And here, an example of a translation that satisfy the validity of the string. Here, in this case, we have an oxygen that would want 3 bonds, 1 to this one and 2, because of this double bond, to this carbon. But here the translated SMILE string actually has the correct bonds, because internally in the derivation, the selfies derivation went into the correct state and knows, okay, in this state it cannot produce a double bond carbon because can only produce 1 more bond. So this potential mistake is fixed here. And also, if we have a fluor somewhere in the center, a fluor can only have 1 bond. So if this fluor has already its bond from here, you add the fluor, then you cannot go, add anything else. So you are in the end state of the derivation, and the rest cannot be translated anymore. So this is correct example.
Mario, can you clarify quickly? So the decoder is writing characters that are not in the representation, but it knows to ignore them. So it's not giving you the no character symbol. It's giving you the double bond C symbol.
So the decoder, this is the selfie string, and the decoder in its derivation creates the semantically correct SMILE string. So in this case, here after the oxygen goes into the state X1, X1 can only produce 1. So the instruction in the state X1, the instruction double bond C in the state X1 produces 1 carbon, and that is what it's doing, a single bond carbon. Was this?
Okay, but there could have been a single bond C instruction.
If it would have a single bond C instruction, it would create the same symbol.
Okay, gotcha, thank you.
Okay, so if there are maybe 3 or 5 minutes, I could show briefly the main idea of the code for the deep dreaming. Is that okay?
That would be great, Mario. Yeah.
Sorry? Good. Okay, perfect. So let me run. I actually found some settings that work very, very quickly. So the idea is we take a huge data set and create the alphabet, and then translate this huge data set into selfies and into one-hot encoding, and train a very simple neural network. It's a fully connected 3-layer with a few hundred neurons. So it gets in this one-hot encoding of the molecule, and it should predict 1 single number. And now let me show you the most important line. Here, this line tells you what the optimizer will act on. Here, the optimizer will act on the model parameters. So this is the standard way how you train a neural network. You adapt the model parameters to fulfill to minimize the loss. So let's copy this line. Okay, so this is how the prediction works, and the most important part, if we finished the training, I think we finished quickly. So here, this is the training loss. This is the test loss. Now we have our fully trained neural network, and we go into the dreaming step. And in the dreaming step, we use exactly the same neural network, but we change where the optimization is working on. And this you can see in this specific line. So before, we were changing the model parameter. in the optimization. But now we don't change the model parameter. So Adam is not working on the model parameter, but Adam is accessing the initial data. So actually the input data. So and they are now changed. And everything else in the whole code is basically exactly the same. This is the only thing that changes in the training. You don't change the model parameters, but you change the input. And here you can see just the sample output. I think it trimmed up 10 molecules. I think we are trimming in the upwards direction into plus. Yes, we trim towards +6. So this is the physical property, and the property is usually between, I think, -3 and +3. So we trim very far into the positive direction. We start with a random molecule, and the trimming changes this molecule to this molecule in this whole steps and goes from 0.5 to plus 2.3. So definitely into the upward direction, right direction. Here again, another random molecule is from -1 to plus 1.8, and so on. Okay, I think this is everything I wanted to show. Just think about it's very easy to do this deep trimming just by thinking what the gradient-based optimizer is acting on. And I was so surprised by seeing, okay, it's only changing one single line, but this is quite cool. Okay, this is basically all I wanted to show in this code. And this code, of course, is we also have a GitHub, and it is called Basitia, and Basitia is the goddess of sleep and hallucination, and so on. So quite fitting there somehow. So this demo you find here.
Thanks, Mario, for the code demonstration, and that's always great job picking names for the repos. There is a couple more questions, maybe a small clarification questions. There is one for Hyun-Wook Young asking about sort of how you're representing implicit, explicit hydrogens and incomplete valencies, like a radical.
Yeah, we can also do it with in a very similar way as it's done in SMILES. We can also add the explicit age. So then this is then just the additional symbol that we have then in our alphabet.
Okay, and then maybe ramping up the discussion. Ole had a really good question, and
okay.
Maybe Dilip, can you make him a panelist, or
Yeah, just
I don't need to translate.
Second. Yeah, happy to do that.
Now you can see me. No, it's a great work. Yeah, just I want to be devil's advocate. So the question is I think for many papers the issue of invalid smiles has been overrated. It's true if you randomly mutate symbols, right? It doesn't work. It's been shown probably like 20 years ago as well. But in practice, no one doing random mutations, and there are other methods. You can do reinforcement learning directly, optimize properties. In the worst case, I can filter out certain invalid molecules, and it's not a big deal. Oh, just a point to start discussion.
Sure, you can do it with many other ways, like what you just said. I would consider this a model-dependent solution, and there are many model-dependent solutions or filtering solutions. One thing that I would mention in this case, actually two things. First, this representation seems to be easier for machines to understand. So here, robustness doesn't come in directly in these translation tasks. Why does the machine find it easier with selfies to translate strings to strings or images to strings compared to smiles. So this is a different application. I had no idea that would be possible. But it's an application of selfies that exploits the structure of selfies in a kind of indirect way. Still don't
May I ask you for a quick
why.
second? Mario, when you said it does better at writing, like they help us as the output or as the input for this?
In the image to string, it is the output. In the string to string, it is both the input and the output. So they trained both directions. So it is easier to read and to write, apparently.
Because the write part will be logical, right? It's the same thing you're doing with the VAE, you're writing, right? So you're writing a sequence from a state, so in a sense, it's kind of the same task.
But here you okay. Yeah, that might be. Wait a moment. Is it the same? Because you don't care about the robustness. You look at in their case they looked both at the Tanimoto similarity but also string-based similarities, and both of them are higher. And in the string-based similarity, you would not use the robustness at all. You would not even need to translate it to a molecule. But yeah, it could be that there's a simple reason behind. But also, as I said, in this example, they use it in both directions, so also for reading.
Then we also have graphs, right?
Sorry?
Then we also have graph-based methods, and then the problem is essentially solved.
What do you mean? What do you mean a graph-based method? You mean the adjacency
Graph
based?
generation, right? 100% valid, no problems.
How can you? Well, you need to encode the domain constraints in some way. How do you do that?
Well, you encode, for example, you can learn balancer rules, and then all your graphs 100% valid molecules.
How do you encode? Okay, if your model is clever enough to learn the rules, yes, sure. But those are domain constraints that are not necessarily clear that it can learn those domain constraints. Just because you use graphs, it's not clear that you get the correct bonding restrictions.
I can send you our paper.
Okay.
It gets 100% valid and like 95% novelty on all this task. But ultimately, I think we all discussing all this all these metrics that are not of prime importance. What's in importance is actual molecule in the vial, right?
Mm-hmm. Mm-hmm.
And yes, who cares if it smiles, selfies, graphs?
Yes,
the ultimate thing is, we want to have an actual molecule as approved as a drug, right? Or as the OLED material or whatever we do applications.
Yes. That's true.
So maybe sorry, Mario, please finish.
Mm-hmm. No, go ahead.
I meant to say that I think having valid molecules is a necessary, or having relatively large proportion of valid molecules is a necessary condition for this working, right? And it didn't work in 2015 when we did the other, 2016 when we did it, because it was 1% valid, right? You're filtering out everything. But I wonder, and Mario, when you showed, your latent space has 100 times more molecules in it, right? But and I think the question that is necessary, and that would be the sufficient part, is, are those molecules from the same distribution as the training data, or is it just a sort of noisy random generator of molecules that sure are valid? They have 27 member ring with all the right number of valencies, but it's sort of outside of because the VA is supposed to learn a distribution, right? Mm-hmm. So the samples should be members of that distribution too. And I wonder if the robustness has allowed you to translate noise better, but it hasn't really sort of learned chemistry.
Yeah, that could be. That it is able to create a lot of different ones. Still, can I mention something that you said 5 minutes ago? You said your models were not able to learn any structure property relations. This is slightly connected. Here, when we use this deep dreaming way of creating a molecule, it is kind of obvious that it has learned, or that this model can learn some structure property relation. Otherwise, you would not see this downward trajectory or this upward trajectory. And because of that, I think you can, even if there are more molecules in your latent space, you can use them really as resources, because it seems that there is a structure property relation very clearly. I mean, it's a very different model. You used two networks and have this intermediate space. Here is basically a direct generation. But I wonder whether this is shouldn't be also possible for VAEs in some way.
Yeah, I think maybe I was being a little bit sort of heavy-handed on, I think this level of performance, I think, builds up on what we used to see, or what has been said. But these molecules are very small. They don't really follow the rules of what I was saying, the distribution, right? Like, was this trained on like zinc or something? What was the training library for this? Zinc?
No, this was the QM9 dataset.
Okay. Well, then okay, fair. QM9 it's indistinguishable, right? Random molecules and QM9 are indistinguishable.
Yes,
so
yes.
We don't know if you're learning the right distribution either. I don't know if folks from the audience, we could go on and on. There was a quick technical question whether there is a query language like Smarts that one could use to query selfies.
Not yet. But if you're interested in, please contact me and we can talk about this further.
And then there was a question from earlier on about the nature of the latent space, in addition to sort of the continuity questions from Chris. It was whether you're learning embeddings for each node sort of independently. Maybe, Darija, you want to clarify a little bit of your question, sort of what you were thinking in terms of learning embeddings for the nodes versus learning embedding for the questions? And maybe Dilip can get you here to the front. Also, we have Pauline and
can you?
I can read. I'm going to read the question literally, and maybe.
Okay. Yes.
Let me see if I can bring it back. Here, it literally says: is the latent space learning simple embeddings for the different nodes in the graph? In other words, how is the latent space model, and how is the grammar of the strings handled?
Every point in the latent space corresponds to one molecule. Where do I have the pictures? Here. Every point corresponds to one complete molecule, not only to some nodes of the molecule. Not sure whether that is the question, actually.
Definitely the part about the nodes versus the full molecule.
No, it is it's a high-dimensional space, and maybe 200-dimensional space, and you decode this high-dimensional point in the space into a string, into first the one-hot encoding, and then translate this into a string. So it contains the whole information. One point contains the whole information of the molecule, and the grammar translation, or the translation one-hot, is done by the decoder, and then you translate it to selfies strings, and then you take selfies to graphs.
I have one about the understandability. I think it's good to a point, and maybe for Alexander too, that I think I can read smiles and like more or less know what the molecule is, like the matrix with the green. Is it like that for selfies? I found a little bit, at the first pass, it seems a little bit counterintuitive, right? To use the same that this meaning of some symbols changes based on the preceding symbol. I think for people to process that, but maybe this is all irrelevant, right? Like, I don't know how many people actually look at smiles, but what do you think?
So we actually did, I gave a talk at some time, and we did some tests, and whether people can read the selfies. Sometimes fail. So I think it helps when you have the stable of the 16 numbers next to you, then you just count. And so I can very likely do this. But then we did the test whether people can read larger smiles, and they also failed. So I think reading them is basically only possible. Either you can read the local neighborhood or only small molecules, but larger ones become really complicated, I think.
Yeah, I can second that. I think the, simple smiles like a benzene, obvious, right? But as soon as you have branching and a lot of rings that have been encoded, branching, things get complicated. I mean, you can read it's like almost genetic code, right? You can read the letters, but you can hardly reconstruct the molecule easily.
Yeah, yes, definitely. When you try, your graph looks so messy, you have no idea what is going on.
And then maybe on the topic of locality, and folks, please stop me. I could go on and on. If anyone has anything, please throw it up. But on the topic of locality, I wonder if there are ways to quantify this, but it looks like selfies is more local, right? Because you open the rings and you already declare when you're going to close them at opening, which I guess could help. Because in a sense, it's like message passing, right? You're sort of telling where it ends already from the beginning. So I wonder if that's what's helping with the translation, and maybe with property prediction too. I know people have gotten smiles for property prediction to work if you give like 1 million training points. And so, but maybe there's a little bit more locality. And I wonder how one could quantify this, right? Sort of, if there is a way to, see how two tokens interact for a certain task, right? Or maybe using attention or the inception that you were doing.
Yeah, that's a very good question. Actually, I got this idea of this locality from a completely different field. There was a huge study of artificial evolution in the early 90s, and they were investigating. It's totally different than a genetic algorithm and these things. You have an artificial environment with instruction sets, and you have codes that can copy themselves, and what happens with them. And they have found actually that they found the number of rules that seems to be very in favor of for those artificial chemistry in the computers. And one of them was actually having information as local as possible. I don't know why that was, but this was studies from the earlier mid-90s. So that was my inspiration to try that.
But is there a way to quantify how much more local it got? I wonder if there was some measure of token locality.
I see you want to, okay, for models themselves. What? Yeah, that's a very good question. Maybe somebody expert in the audience can answer that. So we did not do any tests on this. And how would you perform such a test?
I don't know. Maybe do attribution, right? Where you have a forward model, you can go back and see what tokens mattered and how far away the tokens were for a forward model from smiles and a forward model from
I see.
selfies.
Oh, that's actually, yeah, I see. That's actually a very cool idea.
Right, and you have, right, if it needs two heads looking very far away, that means that you're making it hard for a recurrent encoder, right? To do that.
Oh, this is actually a very cool idea. I will definitely write this down.
We've got a couple more minutes. I don't know, Dilip, if there are any parting notes that you folks typically do.
Yes. So I think, since we're down to the last few minutes, I'll just make an announcement for the next few weeks. The broad theme for the next few weeks is going to be deployment of machine learning methods in the industry. And the first webinar next week will be given by Melanie Sen and Alina Negoita from Volkswagen, with Shailendra Kaushik from General Motors as the session chair. So with that, I want to thank Rafa for taking the time out to chair the session. Thanks a lot, Mario, for such a wonderful webinar. Thanks, everyone, for the interesting discussions and all the questions. So please stay tuned, and thanks for joining, everyone. See you all next week.
Thank you very much.
Thanks,
and
Mario.
thanks for the very interesting questions.
Send my hugs to Alan.
I will. And I will think about your comments, and I will follow up with after I have a good answer to what you said.
No. It was just a thought-provoking question.
No. I like this. This is the most interesting thing where you're forced to think and then you cannot immediately answer. So I like, but yes, I will follow up. Thank you.
Bye.
Yeah. Thanks, everybody.
Thank you. Bye.